Okay, well, very much appreciate the opportunity to speak and share with you what we're doing at Secretome Therapeutics. I'm going to skip around the slides a little bit, so, please tolerate me getting through the deck to the slides that matter for the story I'm about to tell you today. So, Secretome, we're developing novel therapies derived from neonatal cardiac progenitor cells. The reason for us to exist is rather simple. There are several decades of research demonstrating that mesenchymal stem cells, or stem cells generally, have a therapeutic effect. What's often been the Achilles heel for these stem cell development projects, however, has been manufacturing. I know I'm preaching to the choir because anyone at the meeting on the Mesa probably knows that. But in addition to the variability as a substrate for manufacturing, and therefore lot-to-lot variation that's outside the scope of FDA regs, there's variable potency when you derive stem cells from adults. And the reason for both is simple. If I take stem cells from one adult and I grow up 1,000 doses of medicine and then I'm on to the next, phenotypically they're too different. But there's also a lot of variability within the batch that confers on its potency. So both potency and manufacturing have been highly variable and been an Achilles heel for developing stem cells derived from adults. Some researchers and companies have decided to go fully to the other end of the spectrum and get fetal or embryonic stem cells to develop those under the correct scientific premise, of course, that at that stage in development, we're all the same. So it takes away the variability issue, particularly around manufacturing. However, using embryonic or fetal stem cells incurs a whole other host of religious, political, ethical issues that I think will really be problematic to make embryonic or fetal stem cells an FDA-approved medicine. So we're developing neonatal cardiac progenitor cells. These solve both problems. We derive the cells from babies who are born with a congenital heart defect that requires open heart, life-saving surgery within the first 30 days of their life. As part of that surgery, a little bit of the right atrial appendage is removed to cannulate the heart for bypass and thrown away in the operating room. We rescue that tissue, we process the tissue, and we make a master cell bank of neonatal cardiac progenitor cells. We've raised about $27 million, all from family offices, private investors, mostly physicians and biotech executives. We've launched a phase 1 study in a consequence of diabetes called HFpEF, or heart failure with preserved ejection fraction. Our academic collaborator, Emory, launched a phase 1 study in dilated cardiomyopathy yesterday, and I'm going to share with you some of the clinical results. I'm going to move on a little bit. One of the things that's really intriguing about what we're doing is, coming from a small molecule background, there's nothing terribly instructive about having made a lead asset when it comes to small molecules that helps you make the next one. I'm sure there's SAR, et cetera. But with what we're doing, we take our lead drug, STEMO1, it's the native neonatal cardiac progenitor cell. We can knock in a gene, for example, HSF1, and because this is now physically different, and because in vitro studies we showed a difference in potency, we just applied for a patent for this. We can knock in IL-10, which will show up in the cell's secretome, and we can therefore show a physical difference and a difference in potency, and we're applying a patent for STEM04 in the next few weeks. This is something we can do almost ad infinitum, creating a pipeline of stem cell therapies that are broadly anti-inflammatory. This is, as I mentioned, our pipeline. I'm going to skip through these slides in Duchenne muscular dystrophy. Many of you may be aware of Capricor's program and their HOPE-2 data, which is quite strong, and the HOPE-3 data that's coming soon. But we've had an unexpected surprise in the conduct of our study in HFpEF, which is a massive improvement in kidney function, as noted by EGFR. So our development plans, we tend to follow the data, have now really coalesced around developing STEMO1 in different forms of chronic kidney disease. So this is the study design of our ongoing phase 1 study in heart failure with preserved ejection fraction. You can see it's a fairly simple design. There are 2 dose cohorts, either 100 million cells or 200 million cells. They're given twice, on day 0 and day 28, and we measure for efficacy on day 56. In terms of patient disposition, one of the things to keep in mind as I share the data with you is that all of the patients who are valuable for efficacy, patients 1 through 4, were on SGLT2s and GLP-1s. They're on maximal medical therapy. This patient, O5, had calcification of her cardiac valves and therefore was ineligible for efficacy assessments through echo. So we have the safety data, which we'll present, but for O5, we're not presenting efficacy data today. In terms of safety, through 10 infusions, there were 0 hypersensitivity reactions, anaphylaxis, injection site reaction, and there were 0 other drug-related adverse events, which is a nice safety record to start with. These are patients with heart failure. We saw large, consistent increases in 6-minute walk test. We saw large and significant improvements in the Kansas City Cardiology Questionnaire, a patient-reported outcome that basically asked the question, how is your HFpEF today? How is it bothering you? It's a 0 to 100 scale, and all these patients came into the normal range. This patient maxed out at 100. Both of those, by the way, are FDA-approvable endpoints in HFpEF, 6-minute walk, and a patient-reported outcome like the KCCQ. What we saw that was very encouraging was a significant increase in the stroke volume indexed per patient body weight. 3 out of 4 patients had substantial increases in the stroke volume, which, compared to a PRO or 6-minute walk, is not an effort-based measure. It's detected through echo and is highly rigorous. And the increase in stroke volume was driven by an increase in the end diastolic volume. So in heart failure with preserved ejection fraction, just to unpack that term a little bit, of course, ejection fraction is the percent of blood leaving your left ventricle when it beats, and it's typically about 60%. You can have preserved ejection fraction, but still heart failure if that ventricle just becomes smaller. So now in diastole, when you're filling with blood, you're not filling with enough blood, so though 60% is still coming out, that amount of blood is not meeting the body's needs. An important way to address HFpEF and ameliorate the disease is by resolving the diastolic dysfunction. In other words, having that left ventricle relax a little bit more when the blood is filling so that it accepts more blood and can pump more blood. You saw that in the stroke volume, there was an increase in the output, but what's highly encouraging is we're seeing an increase in the input because the end diastolic volume is increasing, suggesting that within 2 months of treatment, we're seeing reverse modeling of the myocardium. NCP and proBNP was fairly normal in the patients. It slightly kind of went down, but not a lot to hang your hat on here. But what was really compelling was the increases in kidney function. For those of you who have a background in CKD, you're aware that really no drug available today for any form of CKD improves eGFR, or estimated glomerular filtration rate. The best we can do is really deviate the downward slope. So this finding of improving kidney function by about 8 points in 2 months is unique, and again, is being produced on top of GLP-1s and SGLT2s. So when we characterize our data relative to the recent entrants in both CKD and HFpEF, the incretins, Wegovy, Mounjaro, et cetera, and the SGLT2s, we see that the improvement in KCCQ lines up pretty well with what the incretins did in their phase 3 pivotal studies compared to placebo. We're seeing the 6-minute walk improvement is very consistent with the treatment arm in these large phase 3 studies that led to approval, and that there is a massive improvement relative to these products, an improvement in kidney function. So I started with manufacturing. Let me just quickly touch on that and the qualities of our cells that lend themselves to being a really uniquely good substrate for manufacturing. So our process is pretty simple. We collect the right atrial appendage, we process it into a master cell bank, convert it to a working cell bank, and we do scale up with our partners at Rooster Bio in Maryland. We've completed a 50-liter manufacturing run at Rooster Bio. We issued a press release on that with Rooster a few days ago. Now we're tech transferring to a CDMO. So our commercial process, though we're still in phase 1, is mostly nailed down. What's interesting about these cells relative to adult cells is that they solve the historic variability issues, right? So if we look at adult cells, this is a study that we conducted and published where we took 5 right atrial appendages from adults. We processed the cardiac progenitor cells, and what you can see is, on a component analysis, they're all over the place, reflecting the heterogeneity of adults. Compared to that, neonates, babies under 30 days old, correcting for race or for gender, there's no variation. These are the neonatal samples. This one was a bit askew, and we learned that that donor's mother was diabetic. Therefore, for the 9 months and 10 days of existence for this organism, it's been 9 months in a milieu of diabetes. So now we know we just don't take donors from mothers who are diabetic. Equally important is that adult cells lose their stem cell phenotype after a limited number of passages. So in our hands, we can take adult cardiac progenitor cells, passage them 4 times, roughly 8 doublings, and they lose c-KIT expression. Not shown here, there we go, is the laser. But not shown here is, we've also looked at telomere length and other attributes around the ability to maintain phenotype and continue reproducing. What we know is these are no longer stem cells after 4 passages. When we look at the neonatal cells, we've taken our cells, we've passaged them out to 20, so exponentially more yield. And we sampled them at passages 3, 10, and 20. We looked at the phenotype that would define them as a neonatal cardiac progenitor cell. They're stable through 20 passages. But in those samples, we also ran them through a disease model, post-MI, looking at improvement in left ventricular ejection fraction, and you can see there's no difference in phenotype or function, regardless of the number of passages. So going back to the slide before, compared to the adult process, where I take a sample from an adult, I can only passage it out so many times and make so many doses, and then I'm on to the next adult who is inherently different. With neonates, I can passage them out up to 20 times, our IND states 15, and then when I'm on to the next donor, that donor is exactly like the one before. We've been granted intellectual property on our lead product, STEM01. We have 2 composition of matter patents protecting our cell, and we have composition of matter patents protecting the secretome that comes from the cell, which we're developing independently for skin burn and other dermatological conditions. Our team is comprised of experienced leaders in the field of biotech. My history is that I was the head of strategy at Reata, which was acquired by Biogen, the chief financial officer at Reneo Pharma, which went public a few years ago, and I conducted the IPO. Our chairman, Henrik Rasmussen, long history in chronic kidney disease as the co-founder and chief medical officer of ZS Pharma, which had their product approved and was acquired by AstraZeneca. And a leadership team that is, in my view, best in class in terms of developing cell therapy. So that's my presentation. I thought I would leave 2 minutes for questions in case there are any, and then if that's okay. Well, I've stunned you into silence, but at least nobody's asleep. So, thank you.
Thank you, Secretome Therapeutics.